Welcome to AP Biology 2.10 about compartmentalization. In this section, the skill set will contain the argumentation. That means you have to predict the causes or the effect of a change or a disruption to one or more components in a biological system. The objectives are: number one, describe the structures of a membrane-bound organelles in eukaryotic cells. The membranes and the membranes, for example, the cell membrane, as well as membrane-bound organelles, mitochondrion, as well as the chloroplast, as well as the lysosomes and the other ones, compartmentalize the intracellular metabolic processes, such as different parts of the cellular respiration, what we are going to learn later on, different parts are going to take place at different parts in the mitochondrion, or the different steps of the photosynthesis, I will use that, photosynthesis, are in different locations in the chloroplast, separated by membranes. These membranes are phospholipid bilayers all around. So we described how they are. Explain how internal membranes and membrane-bound organelles contribute to the compartmentalization of eukaryotic cell functions. I would like to use examples for the explanation. So I would take this lysosome out and make it in a bigger circle inside of the cell, and this is the cytoplasm, and this is the lysosome. Lysosome contains hydrolytic enzymes to break down or chemically digest the engulfed foreign particles or recycle used cellular macromolecules. So what they are doing is hydrolysis, adding water to the macromolecules and break them apart into monomers. If the lysosome's enzymatic content were not surrounded by membrane, in that case, the lysosome would break down or hydrolyze the macromolecules of the cytoplasm. And it would digest totally the entire cell. The membranes separate the different chemical reactions from each other, reaching for higher efficiency in the cells too. For example, the internal pH inside of the lysosome is acidic, so I will just say it's really low. But this facilitates the optimal working pH condition for its hydrolytic enzymes. Sorry about that. On the other hand, this harsh pH would be detrimental to the entire cytosol of the cell because many other cells and cell processes require a more neutral pH. So outside in the cytosol, the pH is neutral. The membrane not only separate the different chemical reactions from each other, but they also serve as a site for metabolic reactions. An example of that is the inner membrane of the mitochondrion. I will take this one and enlarge it. We have the mitochondrion, with the inner membrane. The inner membrane of the mitochondrium contains proteins, different proteins embedded in the membrane, and they are part of the electron transport chain proteins, and also contains an enzyme in here, the ATPase, that makes ATP along the way. The same is true for the chloroplast. We have the chloroplast in here, the thylakoid membranes with the thylakoids, and we have also proteins in here, the electrotransport chain proteins, as well as the ATP synthase enzyme to produce ATP, as well as we have the pigments, the chlorophylls embedded in the thylakoid membrane. Not only the different chemical reactions are separated from each other with this method, but they also increase the surface area to embed many enzymes and proteins. I hope you learned something from the compartmentalization, especially what compartmentalization is and how beneficial it is for the cell, for eukaryotic cells. Have a good day. Bye.